Hello students. Okay, in the last class we discussed about organisms and population. The chapter belongs to ecology. Okay. Now we are going to start a new chapter, that is biotechnology. Right? Actually, this chapter is very important, and this chapter related to the previous chapter, that is molecular basis of inheritance. Molecular basis of inheritance. I think you are studying the chapter present now, molecular basis of inheritance. That is nothing but molecular biology, right? Okay, before going to enter into this chapter, what the importance of this chapter I will explain. What is the importance to read this chapter I will explain. So this is the fourth unit. So fourth unit in your second year. Biotechnology. In this fourth unit, two chapters are there: biotechnology, principles and processes. Second one, biotechnology applications. Biotechnology principles and processes. Second one, biotechnology applications. Okay. Right. Whatever the content present in this chapter, the content is related to the previous topic, that is molecular basis of inheritance. So that's why I am stressing here. Whatever the information given in molecular basis of inheritance, that information you should know perfectly, and you should know the structure of DNA. You should know what is gene, what are the properties of gene. Everything you have to know to learn this chapter, biotechnology. And in the NEET examination, from this unit definitely you will face 4 to 5 questions. Now syllabus is reduced. Okay, 35%. Some chapters removed from your second year syllabus. That's why the remaining chapter has more importance. From remaining chapters they will cover more number of questions. So that's why you have to read this chapter very perfectly. Whatever the chapter present in your second year, all chapters are important and you have to concentrate more on these chapters. The question level also increases the next NEET examination. Why? Because some topics, some chapters removed from your syllabus. So the question set person, the person who set the question paper, he will concentrate more on the remaining chapters. So from each chapter we will get more number of questions also. Previously, up to 2020, from this biotechnology unit, total unit, I am saying total unit, from the 2 lessons, approximately 4 to 5 questions came. Sometimes 6 questions also came. But now the question number also increases. Remember this point. Okay. So let us start the introduction part of biotechnology. So students, write on the side heading biotechnology. Biotechnology. Okay. Right. The term biotechnology was coined by Carl Ehrlich. Right on the point. The term biotechnology coined by Carl Ehrlich. So, Carl Ehrlich, known as father of biotechnology. Father of biotechnology. Who known as father of biotechnology? Carl Ehrlich, the German agriculture engineer, known as father of biotechnology. Why? Because he coined the term biotechnology. So, what is this actually biotechnology? Let us discuss some introduction part here. Later, what actually it contains, we'll discuss. So actually what is this biotechnology? Biotechnology is nothing but it's a branch. It's a branch which combines the techniques with the biology events. Just I am giving a simple definition. Try to understand what I am saying. Biotechnology. Biotechnology. So here what we are doing to get the products which are beneficial for human being, to get that product we are using the biological properties. Try to understand the definition, try to understand what is biotechnology. Everyone say biotechnology, biotechnology, biotechnology. Okay, what is that biotechnology? It's nothing but we are combining the technology field with the biology events to get the products. The products means what type of products which are beneficial for human being. To get the products which are beneficial for human being, we are using the biological events and we are adding some techniques to that biological events to get the product which are beneficial for us. That is simple biotechnology. And in other words, what we can say? Biotechnology is nothing but, see, it's a technique. Biotechnology deals with the techniques which uses microorganisms and enzymes of organisms to produce the products which are beneficial for human being. Just read the definition carefully, students. What I wrote here: biotechnology deals with the techniques which uses microorganisms and their products. The techniques which uses the microorganisms and their products to produce the product which are beneficial for human being. Okay, in simple way, what I am saying here, we are using the microorganisms to get the products from that microorganisms. Simple way, what I am saying, listen carefully. We are using various kinds of the microorganisms and we are using the properties of the microorganisms. We are using the products of the microorganisms to get the beneficial things which are useful for us. That branch is nothing but biotechnology. Simple one. Okay, write down the definition. This is very simple definition for biotechnology. Biotechnology is deal with the techniques which uses microorganisms and their products. Or you can write enzymes of organisms to produce the products which are beneficial for human being. That is simple biotechnology. Right, then I think you understood this definition. Okay, what we are doing here? We are using microorganisms. Simple. We are using microorganisms. And we are using the microorganism properties and we are using the products of microorganisms. the products are beneficial for human being. Simple one. We are using the microorganisms, and what we are using here? We are using the microorganisms properties, and we are using the microorganism products, and which are beneficial for human being. This is nothing but microorganism. With this definition, then what you understood? Simple. Microorganisms are giving some products. We are utilizing their products. This branch is nothing but biotechnology. Right. You got one conclusion, that is we are using microorganisms in biotechnology. But nowadays, the size of biotechnology increased. So in some conditions we are not using even microorganisms. We are not using the products of microorganisms in some cases. Okay, try to understand what I am saying. Just now I told you, by using the products and properties of microorganisms, we are getting some beneficial things that comes under biotechnology. But what I am saying, biotechnology limitations are increased. We are using the we are using this biotechnology for the things in which the microorganisms are not involving. Listen carefully. We are using this biotechnology. To define this biotechnology, we are using some methods. In that methods, we are not using microorganisms. I'll tell you the example, then you will understand what is this actually. Okay, see here. In vitro fertilization. For test tube baby program. Or test tube baby. Right? Next one. Production of DNA vaccine. Next one. Gene therapy. What is gene therapy here? Just correcting the defective gene within a human being. Gene therapy means nothing but here we are correcting the gene. One boy facing some genetic disorder. The genetic disorder means here, what happens here? In that person, the gene will be disturbed. By the disturbing of gene, the person facing some genetic disorder, some diseases, not disease actually, disorder. By correcting the gene, we can cure that disorder. That comes under gene therapy. About gene therapy, you will read in the next chapter, that is applications of biotechnology. We have an example, that is gene therapy. Gene therapy means correcting the defected gene. And production of various antibiotics. All these comes under biotechnology actually. See here one example. In vitro fertilization, we are not using microorganisms. So you have studied in reproductive health various in vitro fertilization methods like GIFT, ZIFT, okay, and ICSI. Some methods are there which help the couple to fertile, to get the child. So in this in vitro fertilization, just we are collecting the ova from mother, we are collecting the sperm from father, and we are allowing for fertilization in a test tube, in the laboratory in vitro fertilization. So if we are not using microorganisms in this condition, in this process, but this process also considered as biotechnology. Remember, this process, even we are not using microorganisms in this process, then also it comes under biotechnology. And remaining production of DNA vaccines, in this we are using microorganisms, and gene therapy. In gene therapy also we are not using microorganisms. Listen carefully. Just we are using some methods to correct the gene, but in this method also we are not using any microorganisms. And production of antibiotics and using various diagnostic methods to detect the disease in the early stage, we are using ELISA, PCR. We are using these techniques. In these techniques also we are not using the microorganisms, and these are also comes under biotechnology. What I am saying here, according to the definition, biotechnology, in this biotechnology we are using the microorganisms. But nowadays the biotechnology limitations are increased, and we are finding some methods which not involve in the microorganisms, that also comes under biotechnology. Please note this point. Okay, let us go for next one. Okay students, hope you understood the definition of biotechnology. Right, before going to learn the concept, we have to discuss about the concept of gene. Why? Because in this chapter completely we are going to deal with the DNA and the genes only. What is the DNA? What is gene? You know everything. Already you have studied in your first year, and in molecular basis of inheritance also we have studied about gene and DNA. But I will give you a short revision for that. Within short I will explain. Okay. And this is helpful for genetics also. Now we are in molecular biology. Exactly. We are learning about the molecular biology. Let us see what is DNA, what is gene actually. Okay. Right. We know that in every organism, whether it is a human being or it may be another animal, or it may be a plant, or it may be a microorganism like bacteria, right? All these organisms are made up of cell. What is the structural and functional unit of organism? That is nothing but cell. Okay, structural and functional unit of organism is cell. Then I am saying functional. Functional means everything comes under this only, functional. This human being is doing something. He is involved in daily activities. He is running, walking, sleeping. Every function depends upon cells which are present in human being. We know that we are made up of some millions of the cells in our body. Like that, animals also made up of the cells, plants also made up of the cells, bacteria also made up of a cell. Then how these organisms are maintaining their activities? How these organisms are maintaining their activities? How they are actually constructed? What is the physical structure of that organism is there, and which decides that physical structure? What part decides that physical structure? That are nothing but cells. अगर थोड़ा अंदर गया हो, cell which contains nucleus, we know that cell contains nucleus, and what nucleus contains? Nucleus contains genetic material. We know this part, everything we know. Genetic material, right students? And this genetic material in the form of DNA and RNA. We know that DNA and RNA, especially if we say that is nothing but DNA is the genetic material. Okay, and this genetic material DNA is present in the form of which here chromosome. कैसे रहता है genetic material DNA? DNA super coil होके एक complex structure बनाते हैं, that is called as chromosome. How is chromosome? Chromosome specific structure is there. Nothing but the DNA only supercoiled. Supercoiled means fold होके एक स्ट्रक्चर, स्पेसिफिक स्ट्रक्चर को बनाते हैं, that is called as chromosome. Okay, if you open a DNA in a cell, in the nucleus, the length of the DNA approximately 2 meters. I'm talking about human being. If you open the DNA structure from a cell, from the nucleus, that DNA length approximately 2 meters. That much of lengthy structure is embedded within the nucleus of each of cell. Then what does the DNA? I'm saying DNA is the genetic material. What it contains? The DNA contains the genes. The DNA, it constitutes. The DNA constitutes genes. We know that. Then tell me students, what are genes? Are they having any specific structure? Genes are having any specific structure? Is there any shape for genes? No. Then what are genes? Genes are nothing but a small portion on the DNA. We know that this is double-stranded DNA. Double-stranded DNA means 2 strands are there. Double-stranded DNA means 2 strands are there. In the double-stranded DNA, okay, this is one strand which constitutes 5 prime to 3 prime, and the strands constitute 3 prime to 5 prime. So just as a small segment of DNA, not a small segment, it's a complete DNA. Just imagine, this is complete DNA. Then what is gene here? Gene is nothing but a small portion or part of the DNA which represents a character. Listen carefully. Gene is a part of the DNA. Gene is a part on the DNA and it represents a character. It is responsible for a character in the organisms. Whatever the height of human being, what the color of human being, what the hair type of human being, okay, everything depends upon the genes. Not only human being, all the characters of all these organisms depends upon the genes only. If you see the plants in the plant kingdom, some plants showing large sized leaves and some plants shows a small sized leaves. Some plants gives red color flowers, some plants give white color flowers, some plants give yellow color flowers, some plants give pink color flowers. What all these are nothing but the characters, and these characters exposed by the genes which are present on the DNA. Which are present on the DNA. Please concentrate, students. What is the meaning of gene here? Gene is a small portion, it's a small part on the DNA which represents a character. What is the meaning of character here? We know the character, height of the person, weight of, not weight, height of the person, color of the person, and the type of posture, body posture, and even the sex also. Male or female, this is also one character. Coming to the plants, size of the plant, and the type of the stem, and the type of the leaves, inflorescence, type of flower, type of fruit, and root system, type of root system, either that is a taproot system or fibrous root system. All these are the characters of a plant. All these characters depends upon the genes which are present on the DNA. Got my point? In animals also, different kinds of animals are there. They are showing different characters, and all these characters depends upon the genes. Remember, whatever the characters we have, and animals have, and plants have, and the microorganisms like bacteria are having, all these characters based on the genes only. Then what are genes? Genes are nothing but a small part or portion on the DNA that represent a character. If you come to bacteria, Some bacteria are round shaped, which we are calling them as cocci. Some bacteria are round shaped cocci. Some bacteria are rod shaped. We are calling them as bacilli. Okay. And this bacteria can tolerate some antibiotics. It cannot tolerate for antibiotic. Just imagine XYZ. We are using an antibiotic XYZ on this bacteria. We are using XYZ antibiotic on this bacteria. This bacteria is able to survive against this antibiotic, but this bacteria is not able to survive against this antibiotic. Then why this character? This is nothing but a character. That means this bacteria has a gene which can show the tolerance against this antibiotic, which can show the resistance against this antibiotic. Got my point? This bacteria, this cocci bacteria has one gene inside the DNA. That gene is nothing but it shows the resistance against the specific antibiotic. So that's why it's able to survive in the presence of this antibiotic. But this is not having that gene. This is not having that gene. This bacteria, which is bacilli, this is not having that gene. In this bacterial DNA, the gene is absent. What happens if the gene is absent? It will not show the resistance character against this antibiotic. So that's why whenever we are growing this bacteria in the presence of this antibiotic, it cannot survive. Why? Because it is not having gene. It is having the gene, that's why it is gaining the character. It possessing the character here. What character? To survive against this antibiotic. Like that, every organism, whatever the character shown by each and every organism, that character is represented by a gene which present on the DNA. I think you understood this concept. What is gene here? If anyone asks, what is gene? Nothing but gene is a part or a portion on the DNA which represents a character. Now tell me, on this DNA, only one gene present or more number of genes present? Tell me the answer. On one DNA, only one gene present or more number of genes present? Right. On one DNA, many number of genes are present. Many genes are present. Okay. You have studied about Human Genome Project in molecular basis of inheritance. In that we have seen that the number of genes in human being. The number of genes in human being. Okay. That we can say that DNA not only accommodate for only one gene. Remember, this part is one gene and this is the another gene. यहाँ से यहाँ तक और एक gene होता और यहाँ से यहाँ तक और एक gene होता और this is another gene. Like that, more number of genes present on the DNA and each gene represents a character. And each gene represents a character. And this thing you have to understand and you have to know to understand this biotechnology, to understand about the genetics also. If you want to understand about genetics also, you have to know this point. Very simple concept. So what is this concept? We have number of cells. In the cells, we have DNA as genetic material, and on the DNA, we are having the genes. The genes are nothing but, the gene does not have any specific structure. Remember, genes are nothing but a specific portion or a part on the DNA which represents a character. Like that, we are having more number of genes on DNA. That's why we are representing more number of characters. Whatever the character we are expressing, all the characters are depends upon genes only. In our body, various kinds of enzymes released. During digestion, various kinds of enzymes released. And every character depends upon enzymes, you know that. Every character also depends upon enzymes. So nothing but here, if you are showing a property, a metabolic property or physiological or physical property you are showing, means that property depends upon enzymes only. Remember, the property depends upon enzymes only. These enzymes are activating one gene to express its character. This enzyme is activating one gene to express its character here. Right? यहाँ पे देखिए मैं एग्जाम्पल बता दूँ. This is one gene, imagine, and this gene representing color of human being. Color of human being, or we can say one plant. एक plant में देखेंगे हम. And by having this gene, the color of the flower is red. By having this gene, the color of the flower is red. And for this gene, the variations are there. By that variation, in some other plant, the color of the flower is yellow. The color of the flower is yellow. And how these variations are coming here? The variations are coming by the presence of enzymes. By the presence of enzymes. What I am saying here, to explain the character for this gene, one enzyme is required. Simple concept. For the expression of this gene character, one enzyme is required. So that's why I am saying all genes are dependent on enzymes. All genes are dependent on enzymes. If the gene has to express the character, means it should be impressed, or it should be what we can say, it should be induced by, it should be promoted by an enzyme. You got my point? So this is the concept. Now we'll go for biotechnology and what is biotechnology, what types are there, we'll go. I think you understood this concept. If you understood this concept only, we can go for further step. Hope you understood. Okay, next one. Okay students, now we'll talk about the definition of biotechnology, which was given by EFB. Sorry. Definition of biotechnology given by EFB. Please note down this very important one. And the full form of this EFB asked so many times in your competitive examination. Write down EFB full form. European Federation of Biotechnology. European Federation of Biotechnology. Then write what is the definition given by EFB for biotechnology. So biotechnology defined by so many scientists. इसका definition so many scientists ने दिया था. Different institution भी इसका definition दिया था. But we have the definition which given by EFB in our syllabus, in our textbook. So that's why we have to read this definition and we have to concentrate. We have to by heart this definition. Why? Because it is very important definition. Write down the definition. European Federation of Biotechnology. The integration between natural science, natural science and organisms, cells, parts, thereof and molecular analogs for products and services. So write down the definitions, very important one, and it will be asked for 2 marks in your examinations. Theory examination, it's a 2 marks question. And if the question asks like this, write the definition of biotechnology or define biotechnology, then also you have to write this definition. If the question asks like this, define biotechnology, then also you have to write this definition. And repeat along with me. Please note down this and repeat along with me. The integration between natural science and organisms, cells, parts thereof, and molecular analogs for products and services. Please repeat again. The European Federation of Biotechnology defined the biotechnology as the integration between natural science and organisms, cells, parts thereof, and molecular analogs for products and services. Whatever the definition we have written in the starting, the same definition, this is also giving same definition, same meaning. We are using microorganisms and their properties to get the products and services. That is the definition of biotechnology. Got my point? Next one. We have some branches of biotechnology. The basic branches are, 2 branches are there. The first one is traditional biotechnology. Traditional biotechnology. And this traditional biotechnology also called as old biotechnology. Old biotechnology, and some scientists told that this old biotechnology is nothing but kitchen biotechnology. It is nothing but kitchen biotechnology. So what I mean to say in this biotechnology, in the old biotechnology, biotechnology is not a present technique. So 5000 BC, I mean to say 10,000 years before. 10,000 years before only this biotechnology process used by human being. But at that time we don't know, the human being don't know that this is biotechnology, this is a branch of biology. They don't know. But what I mean to say here, before 10,000 years only our ancestors started this biotechnology. 10,000 years back only our ancestors used this biotechnology. But then he don't know that is biotechnology. Then in which manner he used biotechnology here? So what is biotechnology? Simply we are using microorganisms to get the product. That is biotechnology. What is the definition of biotechnology? We are using the microorganisms to get the product which are beneficial for human beings. That is biotechnology. In this manner, what is called biotechnology? Preparation of curd. Yes or no? Tell me the students. Preparation of curd comes under biotechnology. That is traditional biotechnology, kitchen biotechnology. So how we are preparing curd? Just we are adding the bacteria. We are adding the bacteria to the milk, and that milk converts into the curd. We are preparing curd. That is traditional biotechnology. And this process used by our ancestors only. We have not invented this process in the present days. The process used by our ancestors, they only told us about this process, but they don't know this is a part in biotechnology. Now we are giving a name for that process, that is nothing but traditional biotechnology. Like that, preparation of bread by fermentation method. Preparation of bread by fermentation method. And preparation of yogurt. Preparation of yogurt. And the production of various kinds of alcohols by fermentation method. Production of various kinds of alcohol by fermentation method. And production of beverages like wine. Okay, production of beverages like wine by fermentation method. All these comes under which one? Traditional biotechnology. So wine produced in the ancestral years only. Wine production started by human being before 10,000 years only. So when the human being civilized, he started the utilization of the things around him. Exactly don't know when he started the production of these beverages, but our ancestor started the production of some alcohols and wines, beer, whiskey, and these things produced by fermentation method by using microorganisms. These are also comes under which one? Traditional biotechnology. And these techniques mean no need to use that much of the things, that much of techniques, that much of hard methods. No need to use these things within these methods. Simple production of curd. How we are doing this? Just adding a bacterial culture to that milk. And yogurt also. Yogurt. How we are preparing it? By adding some bacteria like lactobacillus, something is there. Lactobacillus vulgaris, one bacteria is there that used in Streptococcus species also. Streptococcus species also used to produce the yogurt. And various kinds of the bacteria like Clostridium. Clostridium used to produce alcohols. And molasses use करके alcohol को vinegar को produce करता है especially. Production of vinegar by using molasses by fermentation method by using some bacteria, some fungi, not bacteria, fungi. So different kinds of the products we are taking by different kinds of the methods, by involvement of various kinds of the microorganisms. That all comes under which one here? Traditional biotechnology. So these are all very old techniques, not present techniques. These are all very old techniques. Our ancestors only started these techniques by using microorganisms, nothing but producing the things. And these are all comes under which one here? Traditional biotechnology, also called old biotechnology, also called as kitchen biotechnology. Now one more thing we have to discuss, that is modern biotechnology. The second one is modern biotechnology. So about modern biotechnology, we will continue in the next one, right? Copy these things.